I appreciate the introduction, and it's a privilege to talk with my LifeVantage family here today. I'd like to share my screen about my presentation, so let me take a moment to do that. All right. I think we'll get it going. All right, so the title of my presentation is Biohacking with Nutrigenomics. A systems approach to nutrigenomics, wellness, and aging. And I hope that today's presentation will really turn on the light bulbs and excite you about what kind of science LifeVantage is doing in the field of biohacking. As you already may be aware, nutrigenomics, see if I can get my slide to advance here. There we go. As you may already be aware, nutrigenomics is the science of how food-derived and nutritional products affects your gene expression. Epigenetics is the broader science of how our environment, which includes food, can affect our genes. Remember, food is more than just calories and nutrition. It also acts as a programming code or signal for our body's cells and our instruction set in our DNA. In some of our previous LifeVantage Success Mastery Science talks, we've discussed about how it's better to make antioxidants and chemical substances that our body needs than to take them externally. We can do this by signaling various pathways in our genetic instruction set to turn on the mechanisms that increase these necessary life-giving substances. This is the difference between cellular activation and supplementation. As a quick review, there are currently 3 mechanisms by which we can activate our body's genetic signaling system. The first is the NRF2 signaling pathway, which is a mechanism by which we can reduce our oxidative stress levels by increasing the number of internal antioxidants. This reduces our oxidative stress by 40% within 30 days and as much as 70% within 4 months. This is an incredible feat by itself, but not to stop there. The second method by which we can activate our body is called the NRF1 signaling pathway, where we can increase the ability to support our mitochondria's production and rejuvenation so that we can have more energy and metabolism for the necessary functions that our body needs. And lastly, the third method by which we can activate our body is called the NAD and sirtuin system of activation. It is related to the information theory of aging, which I'm going to discuss in great detail in this presentation. In this talk, I hope to finally reveal the many questions that people have had about these mysterious sirtuin proteins. And so I'll dig deep into the NAD sirtuin theory of aging. The other activation pathways are well covered in previous Success Mastery presentations. You know, articles are all around these days in the major publications that when we signal these genetic pathways in our DNA code, it's essentially like flipping a switch. We do that so that we can feel better and that we can do more. Articles are all around these days in the major publications that suggest that our genetic code potentially would allow us to live as much as 140 years if we didn't suffer from disease. We are actually genetically programmed to live a very long time, but it is the process of aging that affects us all. And so we are very interested in what it takes in order to have a longer health span. There are some pivotal books written about aging recently, and the authors seek a singular theory of aging. These books are fascinating, but are also very technical. I'm going to try to break down some of the major concepts and show how they apply to the science of gene activation and signaling that LifeVantage is involved with today. One unifying theory of aging is that we may suffer from information loss as we get older. When it's talked about in this way, we call it the information theory of aging. Our DNA is such a capable information storage vehicle. All of the world's data could be stored in one room if it was put into a DNA molecule. Our genetic instruction set is actually a form of digital storage, but instead of being made up of digital ones and zeros, it's actually made up of 4 letters, C, G, A, and T. These are the first letters of the chemical base pairs that make up DNA, and therefore our DNA is a base 4 system of information storage. It provides for infinite storage and is inherently stable. DNA has been replicated time and time again over millions of years with rarely a mistake. Where the problem lies is in our epigenome. Epi means above the genome. The epigenome is inherently unstable. That's because it's not digital. It's analog. It has to be analog so that our bodies can respond to the environment and our situations. It allows us to change on the fly. This allows us to adapt. But it's also our Achilles heel. The epigenome can be added to or subtracted from with exposure to the environment, with food, and with time. This then affects how our genes are expressed. In essence, our environment and diet can signal our genes to either turn on or turn off. This is why we say that only 20% of our genes are active at any given time and the rest lie dormant. It's a lot like a DVD disc. The disc on the right is brand new and has all of the digital information and can be read very clearly. However, look at the DVD disc on the left. It's all scratched up. The information is still there, but it's been affected by the environment and with age, and now the information's not readable. Those environmental changes were analog, but the data on the disc was digital. And so it's the same with our genetic code. So how does the surface of the disc or our epigenome get affected? Well, that damage is caused by our exposures to UV light, toxins, buildup of free radicals, and oxidative stress. This chemical alteration essentially scratches our genetic code or even cuts it in places. The same thing is happening to our mitochondria that we use for energy. This is one reason why we want to reduce our oxidative stress and also help our mitochondria. It's helped by NRF2 and NRF1 activation that we've all come to understand. But let's go deeper on this dive with this concept of epigenetic alteration. If you look more closely at the chromosome above, you would actually find out that it consists of a twisted double DNA strand that is wrapped around some small protein spools that are very tightly packed into a fiber that's called chromatin. This is what actually makes up the chromosomes. Taking an even closer look, some of these tiny protein spools, which are called histones, have chemical tags on them called acetyl groups. Acetyl means 2 carbons. These tags are illustrated in red and are placed by enzymes in our bodies and also as a result of our environment. There are also tags that are permanent and can't be removed. These are carbon methyl tags, 1 carbon tags. The more we have of these permanent methyl tags, the more our body seems to be older biochemically. These can actually be measured, and we can allow us to test our body's chemical age even if we are chronologically older or younger. When some temporary acetyl tags are present, the genes are loosely packed, like the ones above, and appear to unwind. And then that code can be read. These are active genes. When the acetyl tags are removed, the DNA is more tightly packed, and the code can't be read. These are inactive genes. In some cases, we don't want genes to be read because they weren't made to be reactivated. If they were to be reactivated, the body would age more quickly. This is where sirtuins come in. The letter SIR in sirtuins means silent information regulator. They help to keep the acetyl tags off so that the genes will be packed more closely and not read, essentially silencing the genes. They also help to support the natural DNA repair process. If sirtuins are always busy repairing the damage by oxidative stress, then they can't be as efficient at their other job of silencing the genes that are supposed to be quiet. This causes our body to use up valuable energy and resources that we need to live. The more permanent methyl tags, though, act like a brake in the railroad tracks, and they prevent the DNA from being read by the train that is supposed to read these genes. Those genes essentially act like a hard stop for our protein-making machinery. They can't be repaired, nor can they be turned back on. So let's look at all of the important jobs that sirtuin proteins do for us. They're involved in all of our cellular functions. Just pay attention to how long the list is. Some of the words you may or may not recognize, but it's not important to memorize them. Just get a feel for it, okay? Supports natural DNA repair, removal of tags, protects telomeres, mitochondrial support, autophagy or cell cleanup, protein synthesis. regulates stem cell depletion, supports apoptosis, supports a healthy, normal inflammatory response, supports glucose regulation, influences mitochondrial biogenesis. That's a lot, isn't it? All of this spells healthy longevity. So in essence, sirtuins are a protein that act like a buffer pad for our scratched or damaged DVD with its genetic code. They are the cellular guardian and housekeeper for us, sort of a CEO. They rejuvenate the surface. They help get rid of the surface noise and scratches. They support the natural restoration of the defects or tags and regulate how well the disc, which is our genetic information, is read. But doesn't a buffer pad need some buffing compound or polish for the pad to be effective? Well, NAD is the polish that sirtuins need. It's the cofactor that's required for sirtuins to do all of that work. NAD stands for nicotinamide adenine dinucleotide, but we'll say NAD from now on, and not NAD. Now, NAD is not only important for sirtuins, but it's also important for other longevity genes and enzymes too. These include suppressing mTOR, which is responsible for the balance between building up and tearing down of things in our body, and also stimulating AMPK, which helps to maintain normal glucose and insulin regulation. As an aside, all of these regulatory genes are also affected by our eating habits and our environmental stress levels. So NAD and sirtuins work together to do all of these vital functions. They include DNA damage control, controlling epigenetic alterations, supporting mitochondrial function, cellular communications, and even helping to support stem cell regulation and a healthy inflammatory response by removing what we call zombie inflammatory cells, which are essentially dead cells that don't know that they're dead. So what have we learned so far? Let's simplify all of this amazing information about how our wonderful body works. It is amazing how involved all of these mechanisms are, but for our purposes, it's just educational background for the simpler things that I'm going to get to. So don't get overwhelmed. Let's review. We have genetic alterations due to oxidative stress and the environment. These alterations cause information loss, aging, and poor function. Sirtuin proteins support the natural cellular aging process. Sirtuin proteins do a lot of good things to protect us. Sirtuin proteins absolutely need to have NAD to do their work. NAD is the key cofactor for other anti-aging mechanisms also. And we want to increase sirtuin protein activity and NAD in order to be healthy and to live a long health span. Bottom line, sirtuins and NAD are good, and we need more. So how can we increase sirtuin protein activity? Caloric restriction and intermittent fasting convince our body that we're in a period of stress and that we need to be in preservation mode. This increases sirtuins. Exercise that stresses us and makes us breathe hard. This also increases sirtuins. Temperature stresses, extremes of cold and heat that push our bodies also increase sirtuins. Some of these are good, but many, such as caloric restriction, are not sustainable by many people in order to be practical to increase sirtuins. Fortunately, nutrigenomic activation of NAD genes also increases sirtuins and is far easier and reliable. The natural history of NAD is that it declines steadily with age, and that sirtuins also decline to about 40% by the time we're 50 and 90 to 98% by the time we're 80. Without NAD, we're dead. We must have it. Sirtuins must have it. NAD is found in every living cell. If a cell doesn't have a source of NAD, it will die within 30 seconds. The left side of the diagram shows what I said before about exercise, dietary changes, and NAD activators being ways to increase sirtuins. Now, how much NAD you have depends on how much you produce versus how much you use up doing the normal daily chores of the body. We can either make NAD from scratch, which takes less energy and time, or we can have what's called a salvage pathway, which allows us to make NAD from used parts. This is faster and more efficient than starting from scratch. Some of the precursors or parts that make up NAD are NR, nicotinamide riboside, and NMN, nicotinic mononucleotide. Just be aware of the names. You don't have to memorize this. All of the steps for NAD are run by enzymes, which tend to be the limiting factor. Although having some extra spare parts doesn't hurt and may actually help a little bit. So here's a diagram about how NR becomes NMN, which finally becomes NAD. NR is on the right and NAD is on the far left. We are going from right to left in this drawing. It's like a conveyor belt at the factory. The red arrows point to the enzymes, which are the rate-limiting steps. It takes 2 steps to get from NR on the right to NAD on the left, and only 1 step to get from NMN in the middle to NAD on the left. We can pile on a bunch of extra NR on the right, but this doesn't help it get converted any faster, especially if the enzyme is just running the conveyor belt at the same old pace. NMN in the middle is a bit closer to the finished product on the left of the production line, but it still has to be processed by the last enzyme to get it to the final product of NAD. What NAD signaling does is to turn on the 2 enzyme steps and make the system work faster and better. And then it can convert the spare parts into the desired product, NAD. This bar graph illustrates that caloric restriction on the left light blue bar increased sirtuin levels by a respectful 94%. When our competitors tried to use NR to try to make more NAD, it only increased sirtuin activity by 2% in 72 hours. Up to this point, it was the only way to get a bump in NAD without caloric restriction and stressing the body. But when we use our NAD synergizer, which signals the 2 enzymes to become more active, the increase was a whopping 100% increase in sirtuin activity in just 24 hours. It was even better than caloric restriction, which increased sirtuin levels. But remember, you had to calorically restrict your diet to get that 94%, and that's not very fun, and it's hard to do for most people. Who needs to figure out all of this diagram to be healthier? When we can have the latest in biohacking technology with Protandim NAD Synergizer, these research scientists figured out all of this stuff so that we don't have to. We can still eat healthfully without starving ourselves. We can exercise and go to the gym as usual, and we can partake of saunas, steam rooms, and cold plunges, all in an effort to increase our sirtuins. But we can go even farther now by taking a nutrigenomic activator to replace caloric restriction or intermittent fasting. So what's the net result when we take Protandim NAD Synergizer? First and foremost, it doubles sirtuin activity within 24 hours. We get a boost in mental and physical energy. We support mood and motivation. And we get improved mental focus and concentration. We support removal of cellular waste. We support healthy DNA function. You remember the scratch DVD and the polish analogy? We maintain a healthy cholesterol level already in the normal range, and we support a healthy inflammatory response using those other natural systems that I said NAD supported. We also support a healthy vascular system. What's not to like here? So remember that this Protandim NAD Synergizer is part 3 of a 3-part system of activating cellular pathways. It's synergistic or working together with the NRF2 Synergizer and the NRF1 Synergizer. This is by design. So a quick review. Protandim NRF2 Synergizer is the cleanup crew. It's your cellular firefighter and sanitation department to clean up damage and put out fires and the flames due to oxidative stress and free radical damage. Protandem NRF1 Synergizer is like the furnace and the energy company. It fires up the powerhouse of the cell, the mitochondria, in all of our cells to produce energy that's needed for life. Finally, the Protandem NAD Synergizer. It's the cell executive officer, or CEO. It tells the cells what to do. It directs, it distributes energy, it mobilizes and utilizes fats, and cleans out the debris and helps replace bad cells. It repairs our genetic material. It sort of acts like your parent. When NAD is depleted, it's like your parent is depleted. Nothing gets done around the house. The dishes don't get done, the hamper doesn't get emptied, and the dirty clothes pile up. Okay, I want to make just a few final points before I finish. You've learned a lot, but now for the icing on the cake. If each of those things does such a good job, what happens when you take them together rather than individually? Well, NRF1 is increased 69% more with all three synergizers present than when NRF1 is used by itself. NRF2 is activated to levels that nobody has ever seen in the history of nutrigenomics, to the tune of a whopping 1,563% above the baseline use of NRF2 alone. Just think of what kind of reductions of oxidative stress are occurring now. Further research studies are currently underway to further document this and patent this trisynergizer effect, and I can't wait. Finally, the NAD is increased by 1,792%. I mean, are you kidding me? That's ridiculous. Look at those two enzymes that were the ones in the NAD salvage pathway again. That conveyor belt is running fast, not slow now. Can you imagine the effect that this will have on sirtuin levels and the other longevity genes that we need in order to have a longer health span? That's what all the talk is about. Here it is, the now patent pending trisynergizer in pack or bottle form. This is why we're so excited. Now, I want to mention the Vitality Stack, which is a biohacking starter kit that won the Bronze Stevie Award in 2018 for the best new health product in the international market out of over 4,000 entries. But notice, it doesn't have the newest Protandem NAD Synergizer. You'll have to add it separately. I'm hoping for this edition in the future. So for now, it's being sold like this, with an added prebiotic in a kit which LifeVantage is calling the Ultimate Stack. I think this is a good way to go, and is personally what I'm doing, because it covers all four bases, all of the pillars of health, the core four, and then some. And it saves you money. So I want to leave you with this super exciting video clip that puts it all together. Wow, right? Listen, in finishing, I want to encourage you to flip the switch on your business. Flip the switches on your family and your friends. Flip the switch on your health. Flip the switch on your genes so that you can feel better and that you can do more. And I want to thank you for this opportunity to share this important landmark science with you today. Skylar, back to you.